You have to learn how is lac operon switched off and how is lac operon switched on. Lac operon is the operon which is found in E. coli bacteria involved in metabolism of lactose. So when the operon is switched off, that means the genes are not expressed, enzymes are not produced because lactose is absent. When the operon is switched on, this means the genes are expressed, structural genes are expressed, forming messenger RNA and then forming polypeptides or enzymes, which are required by the bacterial cell because lactose is present. So this kind of operon is called inducible operon because lactose is an inducer. And switching off and switching on depends on presence or absence of inducer. So please remember, lactose is an inducer. Lac operon is an inducible operon. Now let us learn how lac operon is switched off. This is the operon. These are the three structural genes Z, Y, A. Adjacent to Z, Y, A, this is the operator gene. Adjacent to operator gene, this is the promoter gene. Adjacent to promoter gene, this is the regulatory gene small I, represented by small I, regulatory gene. And this is the promoter of regulatory gene I, PI. This is the promoter of regulatory gene I, and this is the promoter for structural genes Z, Y, A. Now the regulatory gene I transcribes a piece of messenger RNA. This is called repressor mRNA. The regulatory gene I is transcribed into a piece of messenger RNA, a strand of messenger RNA, which is called repressor messenger RNA. This repressor messenger RNA is now translated into a protein which is called repressor. Repressor is a regulatory protein. Now this regulatory protein repressor binds to operator gene. The repressor molecule now binds to operator gene and prevents RNA polymerase to transcribe ZYA because the operator gene in between structural genes and the promoter gene is blocked by the repressor. The RNA polymerase binds to promoter, but it cannot go ahead. It cannot go ahead, it cannot receive, it cannot attain ZYA. So these three genes are not transcribed and there is no translation, no synthesis of enzymes, no synthesis of proteins. So these structural genes in the lac operon remain unexpressed. They are switched off, they are not expressed. Enzymes are not synthesized. This occurs because the repressor binds to operator and blocks the operator gene so that RNA polymerase cannot transcribe ZYA. And this occurs because there is no lactose molecule, there is no inducer molecule. So lac operon is switched off. This is how lac operon is switched off. In absence of inducer, that means the lactose molecule. When lactose is absent, the lac operon is switched off in this way. Now, how is lac operon switched on? How is lac operon switched on? Look at the lac operon once again, ZYA. Adjacent to ZYA, operator gene. Adjacent to operator gene, promoter gene. The RNA polymerase enzyme binds to promoter gene, regulatory gene I. Regulatory gene I transcribes a piece of messenger RNA which is called repressor mRNA. The repressor mRNA is now translated into a regulatory protein which is called repressor. Now lactose is added, which is an inducer. This is the lactose molecule or its isomer allolactose. It is an inducer. It is added, it is introduced into the culture medium of the bacteria. Now this lactose molecule binds to the repressor and forms inducer repressor complex. The structure of the repressor changes and the repressor becomes inactive. The lactose, when present, it binds to repressor, makes it inactive and changes its structure. So this repressor cannot bind to the operator gene. Operator gene is free. The repressor is bound to the lactose. That is why the repressor becomes inactive. It cannot bind to the operator gene. Operator gene is now free. RNA polymerase enzyme binds to promoter. It moves ahead along the operon. Then it binds to ZYA. As a result, RNA polymerase catalyzes transcription of ZYA into a messenger RNA which is called LAC mRNA. This LAC mRNA is now translated. That means the 3 structural genes are expressed. So LAC mRNA is now translated into 3 different proteins which are enzymes: beta-galactosidase, permease, trans-acetylase. So transcription occurs followed by translation. That means ZYA, the structural genes are expressed. That means LAC operon is switched on, producing the enzymes which are required by the bacterial cells because lactose is present. The bacteria require the 3 enzymes for metabolism of lactose. So in presence of lactose, LAC operon is switched on in this manner.